Hello, last section I have to do in cell membranes is 4C, Osmotic Solutions. And here we're going to talk about how water actually dissolves things, and then once the things are dissolved, we'll figure out which way water flows. Remember, water always wants to go where water is not. So the first thing we have to talk about is a solute. Solute is what's being dissolved. So we put sugar in water, we put salt in water, we put rock in water, we put anything in water. Water will dissolve that material. The solvent in this case is water. That's the thing that's doing the dissolving. And a solution is anything that actually has a dissolved solvent inside of it, dissolved solute inside of it. And it basically is the combination of it. So we actually have the solute over here. We have the solvent, which is the water. You can see the water with its bipolar plus and minus areas is able to grab on to things no matter what the charge is and start ripping it apart. And then it'll eventually move until it reaches equilibrium, where the red and blue dots are basically the same. We have a higher concentration there in diffusion. Diffusion will move these things away. We have a higher concentration of blue. Diffusion will move them seemingly towards the red, but that's red that's really moving because the blue has reached homogeneous or an area where it's all the same. Okay, the three types of solutions we're going to talk about. First one is hypertonic. Hypertonic is anytime the solute concentration is greater outside the cell. And so basically the pink things are the solute, and the little blobby things inside of them are free water molecules. And because water is more inside the cell intracellular than it is outside the cell extracellular, water molecules will move out. So this is a hypertonic solution. So which way will water go? It'll move outside the cell. What does it, or how does it move? It's moving through a process called osmosis, which is the diffusion of water, and hypertonic intracellular to hypertonic extracellular. So it's hypo on the inside, it's hyper on the outside. Water always goes from hypo to hyper, so in this case it's moving outside the cell. Hypotonic, this means that we actually have a larger solute concentration inside, more free radical water is outside, and the water will move into it, again moving from a hypo solution to a hyper cell in this case. Which way will water go? Water enters the cell. How does it move? Still moving through osmosis. This is a red blood cell, which actually lives in plasma, which is a high salt content compared to distilled water, and water will move into the cell. It'll go from that hypotonic solution to a hypertonic cell, and it will lyse the cell. And then which way will water go in this sample? Here we have a semi-permeable membrane, same as the cell's membrane. We look on this side and we see lots of little Mickey Mouses. We look on this side, we see very few little Mickey Mouses. Water will move this direction. This makes this the hypo, and this makes the hypertonic. So hypotonic environment will always push water towards the hypertonic. Water always moves from hypo to hyper. Isotonic, which way will water go? Because we have the same material inside as outside, water will both leave and enter the cell. How does it move? Again, it's moving osmosis, and isotonic always moves to isotonic. Just had to say it. So here's the solution. We got red blood cells. Red blood cells actually moving this way. So we have a hypotonic solution. Hypo, water always goes from hypo to hyper. Hyper cell, it actually lyses the cell. Plant cells will swell, but because they have the cell wall going around them, they're not as likely to lyse. Isotonic, water's going from iso to iso. Water's moving out, in and out of the red blood cell, moving in and out of the plant cell. Then in this one we have a hyper, and water always goes from hypo to hyper. So the hypertonic solution actually crenations this cell. In other words, it turns it into like these blobby, pointy sections. Red blood cells are supposed to be able to pick up oxygen from the lung, carbon dioxide from the cells, and then transfer the exact opposite once it gets to the lung. When it's shaped this way, it can't do its job, and you'll get anemic. And what a plant cell does is it actually starts pulling the cell membrane in from the cell wall, and that actually causes the plant to wither, like you see in this case right here. But you give this thing water, pumps up the vacuole, pumps up the cell material inside the cytosol, and it'll actually fill this up, and this plant may come back as long as it hasn't gone too far. Paramecium contractile vacuole, I'm going to show you this one. I'm not going to show you this one. You're welcome to look at it if you want to. And here's a Prezi presentation on cell membrane and transport. It can act as a review for Unit 3. Excuse me, Unit 4. So let's go take a look at a paramecium. We saw in 4B a paramecium flutter around with its cilia and then get eaten. So here is a paramecium. And if you take a look right here, this is a contractile vacuole. And what's going on is water's moving into here. We've got more water outside the cell than we have inside the cell. So water's constantly going inside this paramecium. It gets concentrated in these little valleys you see, and there's another one down there. It gets concentrated in these little valleys or these rays, and it gets pushed into this central vacuole, and then it squeezes the water back out to the environment. So it's constantly being inundated, and then it squeezes the material. I thought that was there it goes. And then here goes the other one. Once it's filled, the vacuole fills, it squeezes out the water. And this movie will just go show you more and more and more of that. So I'm going to go ahead and stop that. I'm going to go back in here, and what I would like to do now is to end the show and end this video. Thank you for stopping by, and we'll see you in Unit 5.